Hello everyone and welcome to Leading the Way in NGS Sample Prep, Twist Bioscience's corporate workshop at AMP 2020. My name is Chris Thorne, I'm the Senior Manager for Field Marketing here at Twist, and I'll be your host for today. Of course, this year we're not able to deliver our workshop in person, but instead we have 3 recorded talks from 3 great speakers. Our first speaker today is Emily Leproust, who is Twist Bioscience's CEO and co-founder and the recent recipient of the 2020 Bio Rosalind Franklin Award, which recognises her as a pioneering woman who's made a significant contribution to the bio-based economy and biotech innovation. Thank you, Emily.
Thank you for the introduction. It is my pleasure to be here with you, although we are all virtual. It has truly been an unprecedented year for all of us, and faced with a global pandemic and general economic uncertainty, we have certainly experienced our share of challenges, and just like you, we persevere. We have been very busy at Twist this year. We've launched a series of new products, including an infectious disease product portfolio. We've built thousands of custom panels, from panels that have 100 probes all the way to panels that have 1.4 million SNP target. And we've reduced with FastHide the workflow time it takes to run the protocol, allowing you to go from sample to sequencer in a single day. Also this year, we expanded our fixed panel to offer a mass exome. We've added new solutions that enable superior performance in a variety of applications. And we collected very useful feedback that is reviewed by our team, by our R&D team, marketing team, to ensure that we are always doing better. As you know, the DNA we make is built on silicon, and our unique silicon writing platform allows us to drive innovation. And we are committed to pushing the boundaries of what is possible, and we're only getting started. Our tools are being used to solve big problems across a variety of industry, from the sustainable production of chemicals to food, and making sure that the world has food security, to the development of new therapeutics, to precision medicine and diagnostics, all the way to storing data in DNA. We can say that our DNA gets around. One example of solving big problems is the tools that we've built to support the pandemic against COVID. 2 major efforts for 2020 have been new panels and controls for screening and surveillance, and off-the-shelf functional antibodies for diagnosis and treatment. In addressing COVID-19, we were able to quickly deploy our teams to develop products to meet the needs of researchers like you by leveraging our DNA synthesis platform. We launched synthetic RNA positive controls used for both NGS and RT-PCR tests, including 13 strands and 13 controls. Our controls have been used in hundreds of labs, as well as listed as a reference material for SARS-CoV-2 on the FDA website. And for those of you that don't know, our controls are the full virus in a tube in RNA form. We also introduced new NGS target enrichment panels used for viral detections and characterization of SARS-CoV-2, as well as for environmental monitoring, surveillance, and to track the evolution of the virus and the strain of origin. Additionally, our respiratory panels allow you to go beyond SARS-CoV-2 to identify other respiratory viruses. We have 15 of them. And our upcoming comprehensive viral panel enables you to capture novel viruses that may jump over to infect humans from non-human species. We've also focused part of our Twist Biopharma antibody discovery capabilities to the fight against coronavirus. And in the past few weeks, we've shown that Twist-developed antibodies are successful at neutralizing the SARS-CoV-2 in live virus neutralization assays. This is incredibly exciting, and we are working to deploy these molecules across a variety of diagnostics and therapeutic applications. These critical new tools are being used to help us forge ahead at fighting coronavirus and are showing you and the world just what we can do with our platform. Our NGS offering is broad, with panels and library prep, and all the buffer enzyme beads that you need to help you achieve your research goals using our best-in-class double-stranded DNA probes. Also on the roadmap, our teams are continuing their effort to provide you with products and solutions, including solutions for oncologies, methylation, and enable the conversion of SNP analysis away from microarray onto NGS. As you may know, we offer an optimized sequencing experience, and the main driver of what we do is the high uniformity of our products, which drives the cost per sample lower. Indeed, because we have a much higher quality of oligo, the oligos are more uniform in concentration, therefore the capture is more uniform, and so you don't have to sequence as much. And actually, we may have to sequence half of what you're used to. So a big saving in sequencing cost. For the development of new customized panel, typically you can do the R&D twice as fast with Twist, because we can produce custom panels in 2 to 3 weeks. And finally, with our fast throughput and fast HYB solution, we've reduced the hybridization time it takes from 16 hours down to 15 minutes, which means that you can go every day from your sample to the sequencer in a day. So those 3 areas, low cost per sample, fast customization, and fast hybridization really helps you do your R&D quickly and then do your production at high throughput and low cost. We're also enabling new applications. For instance, we have a new comprehensive exomes. We have a methylation solution, and as I mentioned earlier, we are helping researchers to convert out of SNP microarrays and use NGS for SNP analysis. We have shown that using Twist plus NovaSeq, you can do SNP analysis at a lower cost than on a microarray. So looking at exomes, we've built a comprehensive exome. So we've taken the core exome that we've had launched and we packed it in with RefSeq. So it's all blended together, you get one tube. And it's the most comprehensive exomes out there and it includes all of cfDNA, RefSeq, and GenCode databases. And we have over 99% coverage across all 3 of those databases. So it's truly comprehensive coverage and the same performance that you've seen with our human core exome, which means that you get best-in-class uniformity, on-target coverage, and lower duplication rate. And so what that means is that you get to get your coverage that you're looking for with less sequencing. We are also building a new area in epigenetics. Our unique silicon writing platform allows us to generate panels that provide robust performance metrics across a range of differentially methylated samples. And in the past, for methylation, fold 80 values of 2.5 or above used to be the standard, and you may have seen 50 to 60% off target. That was the norm for epigenetics with sequencing. But today, with the Twist probes, we can generate very impressive data that offer unprecedented uniformity of as low as 1.6 fold 80 and as high as 90% on target. So again, with the same sequencing budget that you have, you get more uniformity, more deeper coverage, and so just better data. And here you can see some exceptional performance across a wide range of panels and methylation states. We have 0%, 25, 50%, 75, 100% methylation state, and we can totally detect. So it's exceptional performance, and our high sensitivity is achieved even at low input level, which allows us and you to clearly detect target methylation patterns. And we're also able to capture well over both the hypo and hyper methylated regions. So with our technology, we think that we want to make it a no-brainer slow for you to convert from a microarray to NGS, and we're here to partner with you. So we strongly believe that you can challenge us. Come with your microarray project, your SNP microarray project, and we'll be able to show you that by doing NGS plus NovaSeq, you'll get to a lower cost per sample, and you are not stuck to an array content. You can choose the content that you want for your analysis. So bring us your project. We will build you a customized panel, and we'll give you the tool that you need to succeed. So I will say that the future is bright, and with more to come in 2021. Our teams continue to build out the methylation panel and the methylation workflow. We will introduce new tools for microbiology and infectious disease. We'll have enhanced panel designs and continued collaboration to address your biggest research need. So with that, thank you very much, and we look forward to hearing from you soon. Thank you.
Our next speaker today is Gahee Park. Gahee is a research associate in the Department of Oncology at the University of Cambridge and in the Early Detection Programme at the Cancer Research UK Cambridge Centre, where she works in the lab of Dr. Charlie Massey. Her research has been focused on developing a liquid biopsy test by enhancing the analytical performance of NGS-based cell-free DNA analysis. Gahee earned her PhD at the Seoul National University and trained at the Samsung Genome Institute in the Samsung Medical Center. Today, Gahee will be discussing how DNA methylation changes are robust cancer-specific markers and how Twist's targeted methylation solution helped her team achieve greater and higher than previous results, including 3 times better for enrichment and 4 times less expensive than other products used previously. Thank you, Gahee.
Hello, I'm Gahee Park from University of Cambridge, and I'm here on behalf of Cancer Research UK Early Detection Program. First of all, big thank you from everyone at Twist Bioscience. Give us an opportunity to talk about our research. A bit about introduction here. Our lab is under CRUK Early Detection Program and part of ICGC. We believe we can improve survival rate by detecting cancer earlier when the treatment is more effective. To achieve this, we are studying early disease biology and minimally invasive tests using liquid biopsy, especially cell-free DNA. A little bit about cell-free DNA. Cell-free DNA is more fragmented DNA found in the bloodstream. A part of cell-free DNA originates from tumor cells is called circulating tumor DNA. As it contains cancer genomic information, researchers tend to use as molecular biomarker to detect or to monitor the cancer progression. Since previous studies prove circulating tumor DNA analysis is suitable for disease monitoring, we questioned the possibility of detecting cancer early using current circulating tumor DNA analysis. To answer the question, we need to face downside behind this astonishing concept. One is we only have limited number of starting materials. The likelihood of presence of circulating tumor DNA is getting lower by stage, that the possibility of presenting circulating tumor DNA can be below as 0.01%. Since hotspot mutations were well studied throughout the cancer research, people applied hotspot mutations to figure out this circulating tumor DNA analysis can be detected by hotspot-oriented manner. But as we know, there are cancer types that does not have only hotspot mutations. On top of that, the heterogeneity or various types of signal often contributed to biological noises that complex the problem and increase the chance to have false positive or negative result. It directly implies the barriers to technical noise because next-generation sequencing error rate is known to be around 0.1%. Another thing we need to keep in mind is that if we only focus on single mutations, we can't be sure where the mutations come from. Therefore, we need to find a way to link up the missing information between tissue and cell-free DNA. We see the methylation marker as opportunity of detecting cancer-specific and tissue-specific markers from cell-free DNA. Since some cancers, like prostate cancer, DNA methylation is known as stable marker compared to single mutations. We think methylation marker could be a solution for increase the detection sensitivity and decrease the limit of detection by increase the number of target molecules, likewise previously reported using patient-specific panel. However, the gold standard bisulfite conversion method necessary for methylation analysis destroys up to 90% of starting materials. As we are starting with low amount of DNA, we had to sort out conversion method to rescue DNA molecule as much as possible. This is our overall workflow. We take out blood, extract the cell-free DNA, and convert and capture the region of interest to get the differential methylated regions using NGS technology. Since we were finding the best fit into conversion method for cell-free DNA, we tested bisulfite conversion method and enzymatic method for further methylation sequencing analysis. By comparing various bisulfite conversion methods, the enzymatic method performed best by applying circulating tumor DNA or cell-free DNA library preparations. As we know, bisulfite conversion method degrades the nature of DNA. We could find evidence that enzymatic method conserve the nucleosomal positioning pattern that usually can find in cell-free DNA, characteristic of cell-free DNA. Okay. The preservation of cell-free DNA pattern is important in cell-free DNA research area because we now know the fragmentation pattern is different by normal cell-free DNA and cancer cell-free DNA. Moreover, enzymatic method was not only detect individual cytosins accurately, but also covered 15% more CpG sites than bisulfite conversion method. This means enzymatic method increased the opportunity to detect more differentially methylated regions. As we were optimizing our workflow using targeted capture approach, we realized we are still losing a lot of information due to higher number of repetitive regions. We can always sequence deeper, but it will raise the cost. So we wanted to optimize the targeted capture part, and we iterated with Twist Bioscience whether we can recover more information and whether it is more cost-effective. This part is actually part that we fell in love with Twist Bioscience at first sight, but to have fair comparison, we first look at the further enrichment, uniformity, and on and off target regions. Twist showed 3 times better further enrichment, and uniformity was much better, and higher on target rate and less off target rate than other company-based design that I worked for, like using years and years, and I optimized so much. I spent so much time. And the cost, if you think about cost, because I'm from Cambridge, we are very pessimistic, and then we don't have money, so the cost was like 4 times cheaper compared to other companies. It was like, yes, for us. So yeah, and then we look at the region of interest that we are interested in. Blue line is cancer, red line is control samples. We were excited to see we can draw a clear line between the control samples and cancer samples by having less background noise interruption. Trust me, I hate background noise. So we believe that our method will have better detection limit of compared to current method. And we are currently analyzing hundreds samples in cancer detection study. Going forward, we are excited to test impact of assay to improve cancer detection and patient outcomes with a line of sight to analyze large prospective studies through International Alliance for Cancer Detection. Of course, we are going forward with Twist Bioscience panel, Methylation panel. I would like to thank you, everyone, who supported and worked with us. Thank you.
Our final speaker today is Jo Vandesomple. Jo is co-founder and chief scientific officer at Biogazelle, a CRO specializing in high-value genomics applications to support pharmaceutical research, clinical trials, and diagnostic test development. He's also a professor in functional cancer genomics and applied bioinformatics at Ghent University, Belgium, and is a world-renowned expert in the domain of RNA quantification and non-coding RNA. Today, Jo will be talking about how Biogazelle are performing RNA sequencing from liquid biopsies and FFPE samples, and how they're using Twist custom panels in their workflow. Thank you, Jo.
Thanks very much for the kind introduction and for inviting me, presenting some of our work. So Biogazelle is a genomic services company, and we're focusing tremendously on RNA. And we truly believe in the power of RNA as it accurately reflects cellular states. And I sometimes refer to DNA, and please take no offense here, as a static painting. And here, this is Clara, the 5-year-old daughter from Peter Paul Rubens, a famous Flemish painter. And to RNA as real-time still images from a video, and here I just show you a few of these snapshots from a video from a girl, Lotte, a Dutch girl. I'm not sure if some of you saw the video. It's available on YouTube, where the father has documented her every day taking a few snapshots. It's beautiful. While the DNA of the girl has not changed over time, her expression clearly has. And I think it's safe to state that RNA is probably one of the most informative and dynamic information layers to study. So most of you are probably familiar with expressing or studying RNA as fold changes, a little bit more in one sample versus another sample, or a little bit less in one group versus another group. And then you can do differential gene expression analysis for single genes, pathways, beautifully visualized as heat maps, or do even fancy gene set enrichment analysis to understand if a pathway is repressed or activated. And that's all good, but I think it's important to understand that there is also a structural component of RNA. And by doing RNA sequencing, obviously we can deduce and learn from the structural information embedded, encoded in the transcriptome. We can detect mutations, alternative splicing, fusion genes, circular RNAs, etc., if we have the proper processing tools to do so. And at Biogazelle, we definitely have workflows quality controlled to exploit the structural information embedded in the RNA. And it's also safe to say that we have finally entered this era of precision medicine, where we try to provide the right drug to the right patients at the right time. And obviously, molecular tests are crucially important in that. And liquid biopsies are probably becoming, or have already established themselves as the cornerstone of precision medicine for obvious reasons. Easy to obtain, they confer low risk to the patient, they enable serial profiling and really allow us to do longitudinal studies. For those of you in the field of oncology, a liquid biopsy is probably reflecting the entire tumor load compared to a very biased biopsy. And the liquid is full of biomarker potential. There is a flavor for everyone. We have cell-free nucleic acids. We obviously love RNA. DNA is there as well. Circulating tumor cells, extracellular vesicles or exosomes, and tumor-educated platelets. But the challenge of measuring RNA in liquid biopsies is quite harsh, and I will address it in the next slide. I just want to explain in one slide how the RNA is actually ending up in a liquid biopsy. And it's actually a combination of active processes and passive release of RNA. So obviously RNA is a sensitive molecule if there are RNases present, but some of the RNA is quite well protected, bound to proteins or encapsulated in vesicles or platelets or other containers. That's why it is possible to detect RNA in liquid biopsies. There is even some evidence that RNA may be circulating naked, and especially the circularized form through to back splicing, which is pretty robust against exonucleases. RNA may have a communication function like hormones. Whatever its function is in that fluid extracellularly, I think it's important to understand that we can actually exploit RNA as a biomarker, not only from its abundance levels, but also from the structural information encoded in that xRNA. So I already told you the challenges in messenger RNA sequencing of liquids are numerous. It's not too difficult to sequence small microRNAs, but these long messenger RNAs, because they are to some extent degraded or fragmented in the liquid biopsy, we have a lot of unwanted RNA that we don't want to sequence, such as coming from hemoglobin or ribosomal RNA. The input is typically low. We get on average samples that are 0.2 ml, so 200 microliters. You can imagine there's not much nucleic acid in there, although from an RNA perspective, it's at least 10 times as high as DNA. So potentially offering some sensitivity advantages for doing liquid biopsy analysis focusing on RNA. And to complicate matters even to the fullest, the genome is pervasively transcribed and we have sense and antisense overlapping transcripts. So we need library prep methods that can handle all these challenges. The solution in our perspective is probe-based cDNA enrichment or RNA capture sequencing. And we have not invented that technology. It was actually almost a decade ago, where custom enrichments of cDNA fragments has been published by Levin and colleagues, and then gradually the number of genes that were captured from RNA samples increased to actually doing the full exome only 5 years ago, but that was still on high quality and large quantities of RNA. The first clinical sample was actually done in 2014, the first exome capturing on RNA derived from FFPE, and at Biogazelle we were probably the first doing a whole exome capturing of RNA from a liquid biopsy, 200 microliters. So how does the technology really work? Before I do, I just want to indicate that all these capture-focused methods, they repeatedly document that you have higher sensitivity in your readout, you can reveal higher transcriptome complexity, and that this structural information is faithfully maintained in the RNA sequencing data to allow you to do fusion gene analysis and mutation analysis. So we in fact make a double-stranded cDNA library, and by stranded I mean that the transcriptional direction is preserved in the library to make a distinction between the sense and antisense overlapping transcripts. And then we enrich, like we do for exome sequencing at the DNA level, the exons of interest from that complex, highly dynamic range cDNA to enrich the exons of interest for sequencing. It's a pretty laborious workflow, at least in the original implementation. Later in my talk, I will give you a few flavors of more improvements of that workflow. And we actually capture about 400,000 probes to do the full human transcriptome in enrichment. We typically sequence 50 million reads, which shows to be sufficient for a liquid biopsy because there is not much added value in doing so. There always is, but I think it's the law of diminishing returns. And we've done about 1,000 cases so far, mainly in the field of oncology. Bear in mind, in the original implementation of the workflow, it was the double capture round, which we are now abandoning to a single round capture, but I will come back to that later on. This was actually the first data that we generated 3 years ago, where we did matched FFPE plasma and serum from 2 lung cancer and 2 colon cancer patients, and we were actually shocked by the result. In green, you see the number of genes reproducibly detected in FFPE, but also in plasma, here in pink and in dark blue serum, we have about 7,000 to 8,000 genes reproducibly detected in only 200 microliters of fluid, and that was quite encouraging to continue that workflow. The reproducibility is pretty good, as you can see on the left. The trueness or the resolution is very good. 80% of the genes we can detect a 25% difference, so that's really tiny. So the reproducibility and the resolution is pretty high. The gene body coverage is excellent. Please note that we have lower coverage in the UTRs because the probes were not designed to capture UTR, and the cumulative gene diversity indicated that we did not specifically enrich highly abundant genes, so the complexity was quite well in our sequencing data. That's all good and well, but there are still, or there were, limitations to the original target enrichment workflow. To name a few, only protein coding exons of messenger RNAs were enriched, at least in the embodiment of the original implementation. Only human RNA was enriched. Obviously, these two limitations came from the probe set that we commercially acquired. There was no room for customization, whether we wanted more or fewer genes or other genes. I think most importantly, there were no probes against spike-in controls. And these have been crucial to do workflow controls and do some normalization, especially in the field of liquid biopsies. So in the next the next part of my presentation, I want to illustrate with 3 or 4 success cases where we have used Twist Bioscience probes to accommodate some of these challenges. The first example is a custom probe set of about 3,000 probes directed against 170 spike-in RNAs to supplement the messenger RNA probe set. I will give you an example how we apply that to fully document the human biofluid RNA atlas, and also an ongoing study called the extracellular RNA quality control study. Then I will show you an example in which we used a custom probe set of about 450,000 Twist probes against 50,000 human long non-coding RNAs to finish off with a benchmarking study in which we compared the Illumina enrichment with the Twist enrichment. So the Human Biofluid RNA Atlas project is a collaboration with Illumina, Ghent University, and a few other academic centers, in which you want to deeply sequence all human different biofluids. I wasn't even aware of so many biofluids available. There are 22. And we want to deeply sequence and catalog all RNAs present extracellularly in these different fluids. This is actually the work that is coordinated by a talented PhD student, Eva Holstert from Ghent University. You can imagine, as the fluids are so diverse, the RNA concentration is very different, and the integrity of the RNA is quite different in these human biofluids. So we need controls to make data comparable. That's why we spike in a set of 78 so-called sequin RNAs in the fluid prior to extraction, and then we spike also 92 well-known simple spike-in controls, ERCC, to the RNA eluate. And by combined look at spikes that we add prior to extraction and ones that we add after extraction, we can actually accurately quantify the purification efficiency as well as the relative RNA concentration, which is crucial if we start learning what the differences are in RNA amounts in these different biofluids. Of course, I don't have time to discuss the entire outcome of the study, but I just want to give you one outcome, is that there is a huge difference in RNA concentration in these different human biofluids. And this is only possible if you add spike-in RNAs to do that careful quantification. I want to show or draw your attention to aqueous humor. It's an eye fluid, cerebrospinal fluid, and sweat as prototype fluids which have a very low messenger RNA concentration, in contrast to breast milk, seminal plasma, and tears, which have about 1,000-fold higher concentration of RNA per same volume of fluid. What it means, we don't know, but we're actually conducting studies together with key opinion leaders in these different fluids, and we tried to come up with reasons why we would use some fluids for certain diagnostics or for some particular diseases, as well as we try to see if the organ or the tissues that produce or convey the fluid is actually reminiscent in the RNA patterns in the fluid. This is an ongoing study, but I think it's quite fascinating. To finish this one off, I know and I do understand that plasma is a very important liquid biopsy fluid, but I want to just provoke your thoughts is in that plasma may not always be the best fluid, depending, of course, on the disease you're studying. And this is actually the data from matched urine and plasma from prostate cancer patients, which is a logical example, but here we clearly document that if we look at prostate cancer or prostate-specific RNAs, collected from the TCGA databases, that the RNA concentration, again controlled with these spike-in molecules, is much higher in urine compared to plasma. So for some applications, and prostate cancer is perhaps one in which it makes much more sense to look at urine compared to plasma. Now switch gears a little bit. It's the extracellular RNA quality control study. Again, using these 3,000 twist probes to capture the spike in RNAs. And it's actually a very large study, but I will only discuss today the RNA extraction the impact RNA extraction kits have on performance or the RNA pattern output. It's actually a study which consists out of 3 phases. The first phase is we do so-called single-factor pre-experiments in which we want to document the impact that these pre-analytical variables have to come to a phase 2, an almost full factorial, in which we're going to combine these different pre-analytical variables and finally do some real analysis like variance component analysis and a real biomarker study. We have actually finished phase 1. It's already 2 years, and it's a project where 30 people are collaborating, so it's quite hefty. And after summer, we'll start phase 2. As pre-analytical variables are key in liquid biopsy studies, I just want to provide you a sneak preview on the outcome of that study. And of course, all data will be made publicly available. This is the summary of the experimental setup in which we compared 9 different RNA extraction kits for fluids that are marked for fluids at their minimal in yellow and their maximum input volume in dark yellow. I don't know as the real color, I'm colorblind, but you get the point. And then you see that we have different eluate volumes, again, as specified by the kits. I just want to draw your attention that for some kits, this particular one, we have about a 300-fold enrichment of the fluid down to the RNA eluate volume. For other kits, we don't enrich. 100 microliters is the input, and 100 microliter of RNA is the output, so you don't enrich. And you can, of course, imagine that it has a dramatic impact on RNA concentration and yield. And it does. Again, using the spike-in controls, we can actually quantify the amount of RNA that is co-purified or purified in these different kits. There's about a 100-fold difference in RNA concentration from the best performing kit versus the worst performing kit. Of course, this is RNA concentration. You can, of course, concentrate your sample if you wish to, like precipitation. So probably a more fair comparison is yield, but still, depending on the kit you use, so this is RNA eluate volume controlled, there's about 40, 50-fold difference in RNA yield. So if you are into liquid biopsy studies where you have small input volumes, you better make sure you use the kit that will maximize your sample in terms of detection sensitivity. And finally, I think what probably matters a lot, if you do sequencing, you have a high gene yield. So you see that we have, for the worst performing kit, about 2,000 to 3,000 genes reproducibly detected in 200 microliters of plasma versus up to 15,000. So the yield is quite different, and I think we definitely have made our choice which kits we will continue with. Switch gears again. So these are the Twist 3000 probes to enrich spike-in controls. Now we're going to switch to a very customized set of about 500,000 probes geared towards other genes than protein coding genes. It may come as a surprise to some of you, but the majority of the human RNAs or genes actually do not code for proteins. Protein coding genes is this green part, 21,000 genes. Then we have in pink microRNAs, but the vast majority is actually non-coding or long non-coding RNAs. Subdivided into 2 groups, the low confidence ones that you say, yeah, are these really genes? Are they really non-coding? And then in bluish, 50,000 high confident long non-coding RNAs. The problem is that we all love these genes because they have great biomarker potential, they're excellent therapeutic targets, but in classic sequencing workflows, they often escape detection because half of them are not polyadenylated. So we really need a workflow, and enrichment, of course, is perfect to do so, to enrich them from the background, because on top of it, they're also low abundant. So if you work with low abundant RNAs in liquid biopsy, which is already lowly concentrated, that is fragmented, you really need to rely on enrichment to pull out that low signal from the background. So we designed probes against Leakypedia. It's a Ghent University effort. It's probably the most comprehensive human lincRNA catalog. It's published in several papers in nucleic acid research. It resulted in about 450,000 probes, which Twist synthesized for us, targeting about 52,000 long non-coding RNA genes. So obviously we didn't want to sacrifice probes to the most challenging samples, which are liquid biopsies. We first did human brain, and I think we were pretty impressed with the performance. On the left side, you see classic probes against human protein coding genes, but also in pink here now addressing these link RNAs, we have pretty good reproducibility. We detect a lot of genes over a large dynamic range, so we're quite happy and confident to continue for liquid biopsy studies. And these are results, snapshot of our results on plasma, again in green protein coding genes. These are non-twist probes. This is pink twist probes targeting long non-coding RNAs. And I agree with you, the reproducibility is somewhat lower compared to protein coding genes, but bear in mind these are very low abundant genes in only 200 microliters of plasma. So I think we are pretty happy with the performance, especially if you look at the bottom where we say, look, we're only going to keep the data if it's reproducibly detected in 2 technical replicates in this study and above an FPKM value of 1. I think we definitely have data that is pretty useful. We're now conducting large-scale studies to see the power of sequencing long non-coding RNAs in various fluids and see if they can help us do better diagnosis in different human diseases. To finish this one off, just to show you the power of enrichment even for low abundant genes, this is a well-known long non-coding RNA, it's called Gas5, where I show you the coverage in the different exons from plasma samples, 2 replicates here, and you see that the enrichment is pretty good on target, it's relatively uniform, and these arches really denote alternative splicing, and this is very much what we have expected because every single exon of a long non-coding RNA is alternatively spliced, and you really create a lot of diversity in terms of splicing. And this just jumps in your eye from a liquid biopsy in plasma. So it's quite encouraging to continue this type of research. And finally, I want to provide you some of the data and results we generate by comparing, let's say, the latest and the greatest of Twist Bioscience enrichment workflow. I think the differences with what we've done so far, and which I presented, are quite numerous. So we used to work with singleplex. Now we can do an 8-plex with Twist. The hybridization, the washing buffers are different and they're proprietary to Twist. We used to do a dual capture round. Now we rely on single capture. And we used to work with shorter probes, 80 mers, and now we work with 120 mer probes. To really make data very comparable, and I think you can introduce a lot of biases in doing such these kind of benchmarking studies. It's really important that we rely on from the same amount of data. So we did subsampling to 5 million reads, and this is actually work done by Anneline Morlion, another talented PhD student at Ghent University. And we compared platelet-rich plasma with platelet-free plasma. So quite divergent in terms of RNA content, and we had sufficient number of replicates to do a robust assessment of comparing these 2 different workflows. So when we assessed PCR duplication level, we already noticed differences. You may be shocked, and we've seen PCR duplication levels from the previous speaker. Bear in mind, this is extremely low input. So from a liquid biopsy, the duplication levels are high, typically around 80% for platelet-rich plasma, 90% and higher in platelet-free plasma. We know it's there, but you can deal with it. You simply remove the PCR duplicates. But I want to draw your attention to the fact that with Twist, we have about 5-7% lower PCR duplication levels, which is good. And then you may say, well, 5%, that's not a lot. Well, it actually matters a lot, especially in these high duplication levels. For instance, if we then use that information to remove the duplicates and only contain the unique fragments, you see that, for instance, if we look at platelet-free plasma, that we have about 40% more unique reads on target compared to the other enrichment technologies. So it's simply, well, at least for us, this suggests that the enrichment is much more sensitive and we have a larger diversity of molecules and fragments in our ultimate library. And this is important if we want to use that type of data for instance, mutation analysis. The difference is a little bit smaller for platelet-rich, which is a richer transcriptome, but it's still 30% more unique reads comparing this complete Twist workflow with what we have been doing in the past. Also, when we look at how deep we can sequence into the transcriptome and how many genes we detect, we have about 10% more genes with the Twist workflow. And we have about 4-5%, which is actually 50% more deeper sequencing probing into the transcriptome because the enrichment is most likely more efficient and more sensitive. So from a high-level comparison, I think we see similar performance for some of the performance metrics, such as strandedness, repeatability, but we definitely see better performance in terms of PCR duplication levels, gene detection count, and the depth into the transcriptome that we cover with this enrichment workflow. I think most importantly, what we like the Twist Bioscience workflow so much is the high level of customization and the flexibility. So the projects that are now planned or ongoing, because we've seen the successful benchmarking data, is actually a combined human and murine exome enrichment from plasma from murine PDX models. Even more challenging because we have combined human and mouse RNA in only 60 microliters of plasma. Very challenging, and we want to do that combined enrichment and learn which amount of RNA from the human tumor that is implanted into the mouse model is actually leaking, and what can we learn from that. Another one is we're going to completely redo the Linkipedia, so it's Linkipedia 5.2. We're currently finishing the designs and have them synthesized as soon as possible. And then also, because of the level of flexibility, we are doing custom enrichment for rat genes. It's about 1,300 genes, and you might say, well, that's easy, right? And it's even tissue. I just want to draw your attention. These are extremely rare transcripts. Collectively, they consume 0.07% of all the reads if you do a classic RNA sequencing. So we really have to rely on extreme enrichment to address all our sequencing power to these rare genes. So to conclude my contribution to the seminar, I hope I've convinced you that all human biofluids, and there are many, that contain RNA and likely reflecting health and specific disease states. We're actually doing follow-up studies with our collaborators. We have developed and benchmarked sequencing workflows to probe this extracellular RNA, and we offer that as a service. And then the Twist Bioscience enrichment provides a very flexible and high-quality solution for custom target enrichment. Leaves me to thank my colleagues at Biogazelle, Ghent University, and also Illumina and Twist for the valuable contribution and their interest in what we do. My email address is there if you have follow-up questions, so don't be shy to send me an email. Thank you very much for your attention.